These are my additional botany techniques that I know that deal with plants, fungi, and algae. Number 6. Fungi boxes. Last time I showed you how to process fungi in slime molds. Some herbarians might use fungi boxes instead of paper packages. These boxes will have a clear lid and a spongy cushion. I use fungi boxes for more fragile specimens like this bird's nest fungi. Number 5. Epidermal Peels. This is a specific microscopic skill to extract guard cells and trichomes from leaves. First, add the superglue to the slide. Then press your leaf down onto the superglue with a lot of force. Once the superglue has dried, remove the leaf and microscopy it. Number 4. Liquid preservatives. To preserve specimens like algae, I add the alga to a jar and fill it with formalin. Commercial 37% formaldehyde or 100% formalin is diluted with seawater to make a 3 to 5% formalin solution. My buffer, it's 1/4 teaspoon of borax per 1 liter. Number 3. Miscellaneous equipment. The SPAD monitor measures chlorophyll content and micrometers measures the thickness of leaves. The Junior PAM fluorometer measures fluorescence by carrying out the full range of saturation pulse analysis of photosystem 2. Number 2. Miscellaneous Softwares. In most research projects, you probably need to use R and RStudio. My botany program barely involved R, so I know very little about it. I've used 4-peaks for Sanger sequencing analysis to determine the nucleotide sequence of a DNA strand, and I've used Mega 11 once to make a phylogenetic tree, and I used ImageJ once to find the area of a leaf. But yeah. Number 1. Plant DNA extraction, plus PCR and gel electrophoresis. I'll be using the QueChERS plant mini kit protocol on this papaya fruit. First, I collect a small amount of exocarp tissue the size of a paper hole puncher. If I collect more, it could cause inefficient lysis and an increased amount of PCR inhibitors present. This would result in a low yield and purity extractions. Next, I pulverize the papaya with a pestle, add 400 microliters of buffer AP1 and 4 microliters of nRNase A 100 mg per milliliter solution. Vortex mix it to incorporate the reagents and incubate for 10 minutes in a 65 degrees Celsius water bath to release the cells. During incubation, mix 2 to 3 times by inverting the tube. Add 130 milliliters of buffer P3 to the lysates, mix and incubate for 5 minutes on ice to precipitate detergents, proteins, and polysaccharides. Centrifuge the lysate for 5.5 minutes at 14,000 RPM. Pipette the supernatant onto the purple QI shredder spin column placed in a 2 milliliter collection tube and centrifuge for 2.5 minutes at 14,000 RPM. This will remove most precipitates and cell debris, but a small amount will pass through and form a pellet in the collection tube. Discard the purple QI shredder spin column and transfer roughly 450 liters of the flow-through liquid, the liquid that flowed through the column in the collection tube, into a new Eppendorf tube without disturbing the cell debris pellet. Add 1.5 volumes of buffer AW1 to the clear lysate flow-through and mix by pipetting. Mix immediately by gently pipetting at least 5 times. Transfer roughly 650 microliters of the mixture, including any precipitates that may have formed, onto a white DNA-easy mini spin column placed in a 2 milliliter collection tube. Centrifuge for 1.5 minutes at 9,000 RPM. Discard the flow through into the liquid waste container. Keep the spin column and collection tube. We will repeat this step again. Discard the flow through and collection tube. Place the same DNA spin column, which now contains the DNA, into a new 2 milliliter collection tube. Add 500 microliters of buffer AW2 into the spin column and centrifuge for 1.5 minutes at 9,000 RPM. Discard the flow through into the liquid waste container and reuse the collection tube. Add another 500 microliters of buffer AW2 to the same DNA mini spin column and centrifuge for 2.5 minutes at 14,000 RPM to dry the membrane. Discard the flow through and collection tube. After centrifugation, transfer the DNA spin column into a new Eppendorf tube, making sure the column does not come in contact with the flow through liquid because it will result in a carryover of ethanol. Pipette 50 milliliters of buffer AE directly onto the DNA membrane, incubate for 5 minutes at room temperature, centrifuge for 1.5 minutes at 9,000 RPM to elute the DNA from the column. We will repeat this step again. Discard the used DNA spin column and keep the Eppendorf tube that now contains your extracted DNA. Store in -20 degrees Celsius freezer. I don't have footage of the polymerase chain reaction, but basically I calculated a master mix reagent table using the C1V1 equals C2V2 formula. My number of reactions is 4, including 1 DNA sample, 1 positive control, 1 negative control, which is water, and 1 extra. The master mix and the reagents were chilled on ice and thawed with centrifuging. I pipetted the master mix into my tube and centrifuged again. Once my master mix was complete, 23 microliters of master mix were added to each of my 4 PCR vials. An additional 2 microliters of DNA sample, positive control, and negative control was added to the respective vials. PCR vials were flash spin and I didn't need my extra, so I discarded it afterwards. The PCR samples were denatured at 95 degrees Celsius, annealed at 50 degrees Celsius, and elongated at 72 degrees Celsius in the thermocycler. To set up the analysis, purification, and gel electrophoresis, we made a 2.5% agarose gel. In a flask, we added agarose powder and 50 milliliters of 1xTA buffer. We plugged in the flask with a Kim wipe and let the agarose powder soak in the buffer for 5 to 10 minutes before microwaving. Microwave agarose buffer mixture in 30-second 50-second increments, swirling gently during each break. When mixture starts to boil, decrease heat to medium and decrease time to 15-second increments, pausing to swirl the mixture gently whenever it starts to bubble. When agarose has completely dissolved, set flask on a paper towel and remove the chem wipe to cool. Add 2 microliters of green glow, swirl flask gently to incorporate the green glow thoroughly and pop any bubbles. Set up a casting tray with a 15-well comb and use a bubble level. Pour the liquid agarose gel into the casting tray and wait for it to solidify. Put the agarose gel into the electrophoresis chamber and topped with 1 XTA buffer. I got 4 parchment papers, one for the Promega PCR ladder / marker, PCR product, positive control, and negative control. I added 5 microliters of each to the respective parchment papers. I then added 2 microliters of 6x loading dye to each parchment paper. I pipetted my parchment paper solutions into the wells of the electrophoresis chamber. I ran this at 90 volts for 70 minutes. Afterwards, we analyzed it. This right here is my results, and I pretty much stopped here. When I was working with animal samples, I also continued from here and added the exosabbate PCR mixtures with primers to prepare for the Sanger sequencing analysis. Bah!